Hello students, welcome to Dill's Biology, and in this video I will be talking about the comparison between prokaryotic and the eukaryotic cell. In the previous video we talked about the cell organelles, okay, but in this video I will be talking about the distinction or the comparison between the prokaryotic and the eukaryotic cell. So let's start. What is prokaryotic cell? What does it means? If I divide into syllable these forms, that is pro and the karyotic. What does it means? Pro means before or the ancient, and the karyotic or the carrion. Carrion means nucleus. So the ancient nucleus types or the nucleus was ancient. And now what does it means by eukaryotic? Eu and karyotic. What does it means? Eukaryotic means after, or the nucleus is evolved. They have a particular nuclear structure they have. Now I will first start from the prokaryotic cell. That is how the prokaryotic cell are made up. What is the composition of the prokaryotic cell? And in this video I will start from the very deeply. I will start and enter in very deep of the prokaryotic cell. That bacterial cell or the prokaryotic cell have a distinct structure from the eukaryotic cell. That is, they have a cell wall, a cell envelope, and a cytoplasmic membranes. Okay, so they have a extra covering, that is this Glycocalyx. Thereafter, cell wall. After that, cytoplasmic membrane. Okay, and these three protective covering of bacterial cell is known as the cell envelope. And in picture, this is the Glycocalyx. After that, this is the cell wall, and after that, this is in the green color region. This region is membrane or the cytoplasmic membrane. Okay, so now this is a quite complex structure, the complex chemical structure of these three type of protective coverings. Okay, so Glycocalyx consists of two composition, that is, let's erase it. Glycocalyx consists of slime layer and capsule. When the Glycocalyx is actually a gelatinous layer or a loose amorphous viscid secretion of the bacterial cell, and when the Glycocalyx is made up by polysaccharide, is known as slime layer. But when the amino acid as well as the proteins are combined with polysaccharide or the carbohydrate, polysaccharide plus proteins. When combined polysaccharide and proteins, they are known as the capsule. Okay, the glycocalyx layer. What is the job of the glycocalyx layer? Glycocalyx layer helps bacteria or the prokaryotic cell for the ingestion, from the ingestion of the phagocytes, or they helps the bacteria for survive in the a very desiccative environment or the hazardous environment from which the phagocytes are grab the bacterial cell and just destroy it. Okay, so this is the function of the glycocalyx. So now let's move to the cell wall. The bacterial or the prokaryotic cell wall are made up by a chemical substance known as peptidoglycan or peptidoglycan. So when you are going to study about the bacterial cell, you have to know the bacterial cell wall composition, that is the peptidoglycan. Okay, and peptidoglycan is made up of two chemical compounds. The number one is N-acetyl glucosamine and N-acetyl muramic acid. These two type of chemical substance are linked or cross-linked with an amino acid. Okay, so this is the bacterial cell wall. But bacterial cell also have a distinct structure of the cell wall known as gram positive and the gram negative cell wall. So what is gram positive and the gram negative cell wall? Gram positive means or the gram negative. Gram positive means if we apply a gram staining to a bacteria, we apply staining. Okay, and when we remove the stain by decolorization procedure or we apply the acetone to remove the gram stain, thus bacterial cell wall retain the color and known as gram positive. It is a violet colored cell wall. The bacteria show a color of violet or violet color. It is the gram positive. So the example of a gram positive bacteria is present in milk, or sorry, not in milk, in curd, known as lactobacillus trichodes. Lactobacillus. Okay, but what is gram negative bacteria? Gram negative bacterial cell wall, that is, if we apply the gram staining on the bacterium and the bacterium cell wall, it take this staining or when we apply the gram staining, actually, let's clear the view. Apply stain, and when we add the decolorization effect or the acetone for the decolorization, then this bacterial cell wall does not retain the color. Okay, so they look like red. They form the color red. They are known as gram negative bacterium. So The example, what is the example of gram-negative bacteria? That is Escherichia coli or E. coli. Actually, gram-positive bacteria forms a thick and a rigid type of material filled in the peptidoglycan, and in case of gram-negative bacteria or in case of gram-negative bacteria, they are very thin and smooth layer, a smooth deposition in peptidoglycan. Okay, so this is the job of the cell wall. So this is the structure of the cell wall. Now, what is the job of the cell wall? The job of the cell wall is to protect bacteria against desiccation. Okay, they are the protective covering. They helps for the blasting or the collapsing of the bacterial cell. So this is the cell wall, and after that it is come that cell membrane. So this is the cell membrane. Or the bacterial cell membrane. And what is the composition of the bacterial cell membrane? The bacterial cell membrane consists of phospholipid and the proteins. The bacterial cell wall is made up of phospholipid and the protein. Phospholipid and the protein. Okay, so what is the job of the plasma membrane? Plasma membrane also acts as a protective covering after the cell wall. It is the last portion of the cell envelope, and they also contains the enzyme. The cell wall, or sorry, the cell membrane or the cytoplasmic membrane, they have absence of sterol, which is helps the resistance of the antibiotics. From the resistance of the antibiotics, or they help, they have absence of sterol, so they help in resistance against antibiotics. So this is the cell membrane. But there is another structure of the prokaryotic cell which is totally devoid of the eukaryotic cell, that is the mesosome. And this is the mesosome. This portion is the mesosomes. So what is the job of the mesosome? The mesosomes are known as the mitochondria of the prokaryotic cell. Okay, so mesosomes known as the bacterial cell mitochondria. Okay, they are analogous with the mitochondria because they consist of various type of enzymes which is needed for the formation of the cell wall, as well as for enzymes required for the DNA replications also. Okay, so they also have the respiratory enzyme. They done a dozen of job, enzymatic or the biochemical job for bacterial cell. Okay, so this is the mesosome. They are the finger-like projection, or they are simply the appendages evolved from the cell membrane. Okay, now bacterial cell wall also consists of a photosynthetic lamellae. They does not have any type of plastid. They doesn't have the plastid. Okay, they have the photosynthetic lamellae. These are the photosynthetic lamellae. They are the photosynthetic lamellae. They consist of the pigments and enzymes for the light reaction of the photosynthesis. But one exception with the eukaryotic cell. That is, what is the difference? That is, the photosynthetic lamellae have the enzyme for the light reaction only. They have no enzyme for dark reaction. Question frequently comes from this position or this portion. Okay, that is, they does not have enzyme required for the dark reaction of the photosynthesis. Okay, so this is the photosynthetic lamellae. Now, the main organelle of prokaryotic cell, that is the nucleoid. Okay, this is the nuclear material or the nucleoid. The nucleoid does not shows any type of features which is analogous or which is the same, which match with the eukaryotic cell. Eukaryotic cell nucleus, because eukaryotic cell, you know, this is the eukaryotic cell, this is a plant and animal cell. They are the nucleus, or they have the nucleus, consists of nuclear envelope, nuclear matrix, nuclear reticulum, and nucleolus, and the chromosome, DNA or chromatin fiber, which are dense in time of cell division and forms chromosome. They does not show any chromosome. They have not a chromosomal structure. That doesn't have a nucleolus, and that doesn't have a nuclear envelope, which helps nucleus from the environment of the cytoplasm, or the cytoplasmic environment. Okay, they are not distinct for the cytoplasmic membrane. They are embedded in the cytoplasm. Okay, they doesn't have boundary. So this nuclear material or the nucleoid consists of only chromatin, or they have a circular DNA material. This is circular. This is too much hazardous. Sorry. This is a DNA material. Okay. Now, bacterial cell also have extra chromosomal DNA material, or their nuclear material, actually also known as genophore or the bacterial chromosome, it can be called. But there is also another genetic or extra chromosomal genetic material of the bacterial cell or the prokaryotic cell. What is the name of the extra chromosomal material? The name is plasmid. Okay, when it is remain free, this is known as plasmid. But when they are integrated with the bacterial chromosome or the bacterial, sorry, not chromosome, bacterial nucleoid or nucleus, they known as episome. They are known as episome. They are the extra chromosomal material. Bacterial cell also have a structure. They are hair-like appendages or hair-like structure, which is protrude from the cell wall of the prokaryotic cell or the bacterial cell, known as pili. They are made up, their hair-like structure, they are made up by pili proteins. Okay, what is the job of the pili? Pili does not helps bacteria in the movement, in their movement. They are not motile. Okay, they help bacteria to adhering eukaryotic cell or another organism. They help for adhere. Okay, and some pili are also known as the sex pili. Okay, and sex pili, the job of the sex pili is to transfer the DNA or genetic material in the another cell or another organism or the host cell. Okay, so this is the pili. Another structure also protrude from the cell wall. The structure is like that, known as, this is a hair-like structure. This is known as flagella, which helps bacteria in movement. Okay, they are the motile structure. Okay, They made up of flagellin protein. But prokaryotic cell also have another types, that is they have the cytoplasmic inclusions. Cytoplasmic inclusions mean like there is sulfur molecules. They are the lipophosphate. Okay, sulfur molecules occurred in the bacterial cell as globules. Okay, so cytoplasmic inclusions occur as globule like sulfur molecules. Okay, so this is the overview of the prokaryotic cell. The board is too much hazardous and too much ganjam to understand the topic, but I am just trying to understand you or clear the concept as similar as possible, or as much clear as possible. So this is the prokaryotic and the eukaryotic cell. But the major difference of the prokaryotic and the eukaryotic cell is prokaryotic cell does not have a membrane bounded organelles. They have the ribosome. Sorry, I forget about ribosome. Their ribosome have two subunit. One is 50S, another is 30S. Okay, total 70S ribosome. And when these two type of ribosome are work together and adhere with the mRNA for translation, they known as polysomes or the polyribosomes. This structure, the nuclear structure and the ribosome structure and the cell membrane, are common with the eukaryotic cell. Okay, but the as usual or as like eukaryotic cell, the prokaryotic cell does not have a structure, a complete structure. So this is the overview of prokaryotic and the eukaryotic cell. Hope you will like this video, and if you like this video, then please subscribe to my channel to get updates of the more videos. So stay happy and keep learning.